In this presentation, we will explore how cells convert nutrients into usable energy. Specifically, we will examine how glucose is broken down through a series of metabolic pathways to ultimately produce ATP, the primary energy currency of the cell. By the end of this lecture, you should understand the major stages of cellular respiration, where they occur, and how energy is transferred throughout the process. All living cells require energy to survive. Inside every cell, chemical reactions occur in organized chains called metabolic pathways. These pathways either assemble or disassemble the key macromolecules of life. Enzymes play a critical role in these processes. They act as biological catalysts, speeding up chemical reactions so that life can occur at a rate fast enough to sustain the organism. Without enzymes, metabolism would proceed far too slowly to support life. Energy flows through living systems in a continuous cycle. Matter is recycled in the environment from producers to consumers and back again. Cellular respiration breaks down food molecules to release energy, while photosynthetic cells capture energy from the environment and incorporate carbon into living systems. Photosynthesis and cellular respiration are complementary processes. One captures energy and stores it in glucose, while the other releases that stored energy to power cellular activities. Metabolism refers to all the chemical reactions within a living cell that capture, store, or use energy. Living cells rely on two major types of metabolism, catabolism and anabolism. Together, these processes maintain energy balance and sustain life. Catabolism is a series of energy-releasing reactions. During catabolism, complex biomolecules are broken down into smaller components, releasing chemical energy in the process. Anabolism, in contrast, consists of energy-requiring reactions. These reactions build complex biomolecules from smaller organic compounds. Both pathways are interconnected and essential. Cells must continuously break down molecules to release energy and build new molecules to grow and repair. ATP serves as the energy bridge between catabolism and anabolism. ATP fuels many cellular activities, including anabolic reactions that build complex molecules. Much of the energy stored in ATP is contained within its phosphate bonds. When ATP loses its terminal phosphate group and becomes ADP, energy is released. Catabolic pathways regenerate ATP, ensuring a continuous supply of energy for the cell's needs. Energy is extracted from food through oxidation-reduction reactions, also called redox reactions. Oxidation refers to the loss of electrons. Reduction refers to the gain of electrons. These reactions always occur together. When one molecule is oxidized, another is reduced. Redox reactions are central to cellular respiration because they allow energy stored in electrons to be transferred and captured. In cellular respiration, oxygen-dependent catabolic pathways extract energy from glucose to produce ATP. During this process, glucose is oxidized as it loses electrons. In contrast, photosynthesis is an anabolic pathway in which carbon dioxide is reduced by gaining electrons and hydrogen atoms to form glucose. Together, these processes demonstrate how redox reactions drive energy transformation in living systems. Enzymes often function in sequences known as metabolic pathways. In these pathways, the product of one reaction becomes the substrate for the next. This organization allows multi-step pathways to proceed rapidly and efficiently. Because enzymes are positioned close together and reactions are tightly coordinated, cells can regulate energy production with precision. Energy carriers are organic molecules that receive, store, and deliver energy within the cell. The most widely used energy carrier is ATP, adenosine triphosphate. ATP releases energy when it loses its terminal phosphate group, becoming ADP. This conversion allows energy to be transferred quickly and efficiently to cellular processes. In addition to ATP, cells use specialized electron carriers such as NAD+ and FAD+. These molecules store energy in loosely bound electrons and hydrogen atoms. During catabolic pathways, NAD+ and FAD+ pick up electrons and hydrogen atoms, becoming NAD and FAD2. These reduced carriers will later deliver high-energy electrons to the electron transport chain, where large amounts of ATP are generated. Photosynthesis and cellular respiration are closely connected processes. Only producers, such as plants, algae, and certain bacteria, can carry out photosynthesis. They use ingredients from their non-living environment, including carbon dioxide, water, and light energy. In contrast, all eukaryotes and most prokaryotes perform cellular respiration. Cellular respiration allows organisms to extract chemical energy from the food they consume. Together, these processes drive the flow of energy through ecosystems. Cellular respiration requires oxygen to break down food molecules. The energy released from these molecules is used to generate ATP. As glucose is broken down, carbon dioxide and water are produced as byproducts. This process converts the chemical energy stored in sugars into usable cellular energy. Cellular respiration occurs in three major stages. First, glycolysis takes place in the cytosol. Second, the citric acid cycle, also known as the Krebs cycle, occurs in the mitochondria. Third, oxidative phosphorylation takes place in the mitochondria. Among these stages, oxidative phosphorylation produces the largest amount of ATP. Glycolysis is the first step in breaking down glucose. It occurs in the cytosol and does not require oxygen. During glycolysis, enzyme-catalyzed reactions convert glucose into a 6-carbon intermediate, which is then split into smaller molecules. Ultimately, glucose is converted into pyruvate, a 3-carbon organic acid. Pyruvate then enters the mitochondria for the next stages of cellular respiration. Glycolysis does not require oxygen. It is an anaerobic process. When oxygen is unavailable, the pyruvate and NAD produced during glycolysis remain in the cytosol. Post-glycolytic reactions convert pyruvate and NAD into other molecules, such as alcohol or lactic acid. This process allows glycolysis to continue producing ATP, even in the absence of oxygen. In alcoholic fermentation, anaerobic yeast convert pyruvate into ethanol, releasing carbon dioxide gas. In lactic acid fermentation, pyruvate is converted into lactic acid. Lactic acid fermentation occurs in severely taxed muscle cells when oxygen levels are insufficient. Although fermentation allows ATP production to continue, it produces far less ATP than aerobic respiration. The second step of cellular respiration occurs in the mitochondria and is an aerobic process. Highly aerobic tissues, such as muscle, contain large numbers of mitochondria and have a rich blood supply. This ensures that oxygen is delivered efficiently for ATP production. The mitochondrion is therefore the primary site of energy production in most eukaryotic cells. Before entering the citric acid cycle, pyruvate must be converted into acetyl-CoA. Through a series of reactions, the citric acid cycle produces ATP, NAD, and FAD2. These energy carriers will deliver high-energy electrons to the final stage of cellular respiration. Carbon dioxide is released as a byproduct during this cycle. The Krebs cycle takes place in the mitochondrial fluid, known as the matrix. It uses oxygen indirectly through the electron transport chain. During this stage, pyruvic acid is broken down into carbon dioxide. High-energy electrons are produced and transferred to NAD and FAD2. A small amount of ATP is also generated. The majority of ATP, however, will be produced in the final stage, oxidative phosphorylation. Oxidative phosphorylation produces the largest amount of ATP during cellular respiration. It is the final stage of cellular respiration and takes place in the many folds, called cristae, of the inner mitochondrial membrane. Because the inner membrane has extensive surface area, it can support a large number of electron transport proteins and ATP synthase enzymes. This structural organization allows cells to maximize ATP production. Energy carriers produced during the Krebs cycle, specifically NAD and FAD2, donate their high-energy electrons to the electron transport chain. As electrons move down the chain, energy is released step by step. This released energy is used to pump protons across the inner mitochondrial membrane, creating a proton gradient. This gradient represents stored potential energy that will be used to synthesize ATP. Protons flow back across the membrane through a protein channel called ATP synthase. The movement of protons through ATP synthase activates enzymes that catalyze the phosphorylation of ADP to form ATP. Electrons traveling down the electron transport chain are eventually accepted by oxygen and hydrogen ions to form water. Overall, cellular respiration yields approximately 30 to 32 ATP molecules per molecule of glucose. This high ATP output makes aerobic respiration extremely efficient. Cellular respiration is not limited to glucose. Carbohydrates are broken down into glucose, which enters glycolysis. Proteins are broken down into amino acids, which can enter metabolic pathways at different points. Fats are broken down into fatty acids, which are converted into molecules that enter the citric acid cycle. Regardless of the starting nutrient, carbohydrates, fats, or proteins, the goal remains the same: to generate high-energy electron carriers that feed into the electron transport chain, producing ATP.